First of all, I'd like to thank the organizers, Watanabe Sensei, Biru Sensei, and others for organizing this transdisciplinary workshop and symposium, which is really amazing, and I am very much pleased to be here. So, in upcoming 30 minutes, my topic is something like life beneath the seafloor. So going back to 1994, the Professor John Parks at Bristol University and his colleagues reported a striking paper in Nature reporting that the numerous number of microbial cells are present, present in marine sediment down to over 500 meters below the seafloor. The cell concentration is over 10 to the 5th to 10 to the 6th per cubic centimeter of sediment. And the concentration is logarithmically decreased with increasing depth. And they mentioned in the, describing the Nature paper that these findings significantly extend the depth of the marine biosphere and will contribute to global biogeochemical cycle, especially in carbon, long after the burial. Nevertheless, at that time, or still now, the bottom of the biosphere still not yet explored. To date, a total microbial cell abundance in marine sediment is estimated to be 2.9 times 10 to the 29 cells, corresponding to 4 gigaton of biomass carbon on Earth. The concentration of cells, microbial cells in organic rich sediment at high sedimentation lake area along the continental margin are much higher than the organic poor sediment at low sedimentation lake area in the open oceans, which suggests that the surface and subsurface biosphere linked tightly together. So in 2010, the Professor Steve Daunt at URI and I organized the Integrated Ocean Dream Program, Expedition 329, using geodesic resolution, and explored the most oligotrophic ocean in the world, the South Pacific gyre. So we drilled 7 sites along the north and south transect across the gyre heart, and confirmed that the roughly 10 to the 3rd to 10 to the 4th cells are present throughout the sediment column from the seafloor down to the basement. This is very important because even though the sediment contains very low organic matter, maybe TOC is less than 0.02%, there's some sizable cells throughout the sediment column, suggesting that there is no limit of open ocean sedimentary biosphere. So we also confirmed the penetration of oxygen throughout the sediment column from the seafloor down to the basement. So this blue area represents aerobic sedimentary habitat, whereas white area is anaerobic. So this aerobic subseafloor biosphere comprises up to 37% of the global seafloor. Another important aspect in the sedimentary biosphere is presence of radioactive elements, as we discussed in this morning. The uranium, thorium, potassium, and such radioactive elements react with pore water and generate hydrogen peroxide, hydrogen, and electron, and this is so-called radiolysis, which may occur during the 4 billion years history and support microbial life. And the Justin Savage, graduate student at the Steve Daunt lab at URI, published a Nature Communications paper, which is a very good paper, actually, the Nature Communications last year. And she calculated the radiolytic hydrogen production and made a global map, and concluded that the water radiolysis is the principal source of biologically accessible energy for microbial community in marine sediment, especially older than a few million years, because the surface sediment contains some consumable energy. Then the question is, what kind of microorganisms living there? So we extracted eDNA, the environmental DNA, from deeply frozen core sample, more than 300, collected from 40 different sites all over the world by scientific ocean drilling. And that we analyze under the condition of the same subsampling and analytical approach. So first, we tackle an issue of so what about the abundance of two domains of life, archaea and bacteria. It has been long debate, what is archaea or bacteria, which is abundant. But we use a more sophisticated molecular approach using a microfluidic digital PCR technique and estimated that archaea cells constitute 37.3% of the total microbial cells, corresponding to 1.1 times 10 to the 29 cells in the global sedimentary biosphere. So interestingly, the marginal sediment is much higher archaea population than those in open ocean sediment, which is interesting. Why archaea is abundant in marginal sediment? Probably the archaea is sensitive to the pressure because the open ocean is mostly abyssal plain over 4,000 meters, or this difference reflects the difference of ecosystem type, aerobic and anaerobic microbial ecosystem. Then we amplify the 16S lysosomal RNA genes and study the taxonomic composition of those aerobic and anaerobic sedimentary biosphere and compared to those community structure in topsoil and seawater. So anoxic sediment and oxic sediment, the community structures are slightly different, but there are some shared microorganisms. However, these sedimentary biospheres are markedly different to those in topsoil and seawater, which is actually the surface world on our planet. Then the community structure is strongly influenced by metadata, environmental factors such as presence or absence of oxygen, sulfate, TOC concentration, and also this community structure is somewhat associated with lithological characteristics. So next, we estimated the diversity richness, how many species in there. That we use 5 different models, and asymptotic relation model is best described the species diversity and estimated to be 7.8 times 10 to the third for archaea, and 3.2 times 10 to the fourth for bacteria, which is amazing because it's very roughly similar level of global microbial richness, and such as topsoil and the seawater. I think this is, when I saw this species richness, I'm a little bit surprised at why it is so similar, because surface world is very much energy rich environment, whereas subsurface is energy limited. I don't know why, but the compiled result indicates that the bacteria are more diverse than archaea in Earth's global biosphere, including surface and subsurface. This is the most important conclusion in this study. The question is, what are the ecological role and the limits of deep subseafloor sedimentary biosphere, especially in the continental margin sediment? In 2002, it's Professor Kyle Henrich and I organized the IODP expedition CC7, the deep corvette biosphere of Shimokita. This was the first deep biosphere dedicated laser drilling expedition using the drilling vessel Chikyu, and the target is deeply buried corvette in the ocean over 2 kilometers below the seafloor. The drilling site is situated here, roughly 120 kilometers offshore the Shimokita Peninsula, and they're located in the fork basin of the Hidaka Trough, and the water depth is 1,200 meters, and the corvette is at around 2 kilometers. Then, in this slide, the panel A shows the cell concentration profile along with depth, and we expected that the cell concentration is decreasing with increasing depth along the global regression line. However, the data shows that the cell concentration is sharply decreased at around 1.2 to 1.5 kilometers. Nevertheless, we see some biosignatures down to the bottom, so 2.5 kilometers. Nevertheless, we see some cell proliferation at corvette up to 10 to the 4th cells, which is probably due to the energy availability within the corvette. Panel B shows the methane the carbon isotopic composition of methane and the hydrogen isotopic composition of methane shows the occurrence of hydrogenotropic CO2 reducing methanogenesis. The carbon isotope of methane is always lighter than -60 per mole, and the hydrogen isotopic composition of methane is at around -200 per mole. So this thing, the CO2 reducing hydrogenotropic methanogenesis instead of acid-clastic methanogenesis. When we see the C1-C2 ratio, we see some nice peak around here, the coal bed, and also the carbon isotopic composition of CO2, the green plots also enriched, they're getting heavy, also suggesting that the occurrence of isotopic fractionation mediated by microbial activity. There are many geochemical and microbiological evidence indicates that the microbial life is present even down to 2.5 kilometers below the ocean floor and play an important role in biogeochemical carbon cycling. We extracted DNA and studied the taxonomic composition of indigenous microbial communities. And markedly, the taxonomic composition is clearly different between shallow and deep microbial communities. In shallow sediment, for example, the 50% of the community structure consists of chloroflexi, followed by Atribacteria, OD1, and some others. But in deep microbial communities, it consists of actinobacteria, proteobacteria, Firmicutes, and chloroflexi is only a few percent. It's very different. And these deep microbial communities resemble organotrophic community in forest soils. So when we traded such a community structure with statistic method based on Barry crisis dissimilarity analysis, it nicely shows that the community structure is somewhat associated with the depositional history from terrestrial, lacustrine to coastal, and hemipelagic, because like 15 to 20 million years ago, the Japan Sea is spreading, and then the Shimokita Peninsula that on the coast is subsiding. So that this is the history of subsiding from terrestrial to deep sea. So this suggests that terrestrial sediment retain indigenous microbial community over 20 million years after burial in the seabed, which is amazing. And the question is, if we could cultivate those deeply buried microorganisms. The shipboard scientist Hiro Imachi used the down-hanging sponge DHS flow-through bioreactor system, and he operated this system over 100 days or more, and then successfully enriched the microbial communities. So during the operation, the deeply buried microbes start reviving like zombie, and we see the somewhat healthy cells, but all cells are digging into the coal particles, and because they are very much starved and hungry. And then this enrichment culture provide a very important and excellent inoculum source for isolating some new healthy subseafloor microbiome, which includes the CO2 reducing hydrogenotropic methanogens such as Methanobacterium relatives. Another shipboard scientist of the Expedition 37, Chan Hong Liu, at the Nanjing University, did a wonderful job. So he cultivated many fungi from coal and shellbed. This is a eukaryote instead of prokaryote. The eukaryotic fungi, the isolated from the 2 kilometer deep coal and shellbed. And one of the representative species, Schizophyllum commune, which is a typical abundant wood decay fungi. And we sequence the genome, and the genome structure of subseafloor fungal isolates, including this isolate, show the 3 orders of magnitude lower nucleotide diversity, substitution, and homologous recombination rates than other terrestrial type strains. This is very important because these sedimentary fungi have experienced an evolutionary status. Evolution is stopped in the deep subseafloor because there is no, the driving energy. So that the most likely, these fungi are buried in sediment as spores and then persist in sediment for over millions of years. So this slide shows the compilation of cell abundance accumulated datasets so far, and it's clear that organic buried-oriented matter concentration and depth functions such as temperature, pressure, and physical properties like porosity, permeability are important factor, but limits of life that define the habitable zone remain poorly constrained. Shippo scientist Wataru Tanikawa at JAMSTEC KCC studies geophysical limits to the deep corbett biosphere. And he found somewhat positive correlation between cell concentration and porosity and permeability, and figured out that the small spores less than 0.2 micron may act as a barrier to water and energy-yielding substrate for deep microbial life. Of course, temperature is a critical factor for microbial persistence. This colored plot shows the biomolecule damage rates, such as DNA hydrolysis and amino acid racemization. And you see this damage rate is sharply increased at around 55 to 60 degrees C, like a boiling egg, you can imagine. So this is very important because the energy requirement for biomolecule damage fixation increases with temperature in situ. So that our hypothesis is that the cell balance and the energy supply should be balanced in the deep biosphere. In order to test this hypothesis, we organized against the IODP Expedition 370 in 2016, the temperature limits in the deep biosphere of Muroto, so-called T-limit project. The drilling site is situated here, the protothrust zone of the accretion prism in the Nankai Trough, the 125 kilometer of the Cape Muroto, and the water depth is roughly 4.7 kilometers, and the drilling depth is 1.2 kilometers. So this area has previously drilled by the ODP, multiple ODP expedition like ODP like 130, 190, and it has been known that this area is particularly high thermal gradient, so that if we drill down to the basement, the in-situ temperature should be around 120 degrees C. So the wind confirms that the bottom temperature is 120 degrees C. And then this slide shows the cell abundance, cell concentration, and depth. And so we see the cell concentration is decreasing with depth again, but reached at the boundary, the least the detection limit at around 400 meters at 55 degrees C. This indicates that the deep buried cyclophilic to mesophilic microbial community is sensitive to the surrounding temperature and then decreased with depth. Nevertheless, below this boundary, we see some microbial population indicating the occurrence of thermophilic microorganisms in those horizons. So interestingly, we see some lifeless zone environment at around 600 meters below the seafloor and 900 to 1,000 meters below the seafloor. So this horizon, at around 600 meters below the seafloor at 70 degrees C, there are some the consolidated volcanic tuff layers. So the Tanikawa-san, they said it might be some the porosity is much less than 0.2, so there's a barrier of energy supply for microbiomes. So this horizon between 900 to 1,000 meters at around temperature is roughly 100 degrees C. This is called the overpressure zone, so mechanically weak, and we observe some hydrothermally altered minerals like barite, for example, and some hydrothermal fluid transiently, transported to here and sterilize the microbial community. This is the hypothesis. In any case, we see some lifeless zone. However, surprisingly, below this lifeless zone environment, the cell concentration is starting increasing again, and we see 400 cells per cubic centimeter at the bottom. This indicates the occurrence of a hyperthermophic microbial community in this environment. So when we look at the geochemical profiles, it is clear that the upper sediment shallower than 700 meters below the seafloor is a biogenic methane, and the deeper zone is thermogenic methane. There's some flux from sulfate, roughly 6 millimolar from the basaltic aquifer, and we see the sulfate methane transition zone at around 750 meters below the seafloor at 82 degrees C. Interestingly, so when we see the carbon isotope profile here, the white dots, so we see some peak here associated with a sulfate methane transition zone. The carbon isotope is getting temporal getting heavy. It's suggesting the occurrence of thermophilic microbial anaerobic oxidation of methane here. Also interestingly, the C1-C2 ratio is slightly increased, and the carbon isotopic composition of methane is getting lighter here, also suggests that the occurrence of hyperthermophic methanogenesis in this environment. So when we look at the potential methanogenesis rate using the carbon-14C and the radiotracer method done by the Professor Tina Trude at UCLA and Queens, and we see some mesophilic methanogenesis activity here and thermophilic methanogenesis here. But also, so we observed some hyperthermophic methanogenesis activity here, which is amazing, which supports the notion that the occurrence of hyperthermophic ecosystem here. The panel C shows the acetate. The acetate is a very important and good intermediate for microbiome because microbes can produce acetate and consume acetate. Therefore, the acetate concentration in upper sediment is relatively low, and the carbon isotopic composition of acetate is fluctuated. However, at around 600 meters, the acetate concentration is sharply increased, and the acetate isotope is getting, some stable. The acetate concentration is accumulated up to 12 millimolar, which is something like vinegar, which is amazing. Actually, only a very small portion of pour water, so that I couldn't leak, but it's very high concentration of acetate accumulated here. Interestingly, the acetate concentration is decreased here at the high temperature zone, and then acetate carbon isotope is enriched in 13C, getting heavy, suggesting that the occurrence of acetate consumption associated with methanogenesis. So that's such an activity measurement that we can calculate the long cell-specific carbon turnover and the biomass carbon turnover time, and high temperature carbon turnover time is much less than cooler sediment, sometimes less than 1 week or 1 day, which is compatible to some laboratory cultures, which is amazing. And this data, real data consistent with the notion that the high energy demand needed to withstand and repair thermal biomolecule damage. Okay, so there are some significant questions remain to be addressed. How is the deep biosphere evolution associated with plate tectonics and mantle convection? What are the roles of deep crustal biosphere in inertial element cycling? Is plate tectonics required for emergence of life? And why the planet Earth is habitable? So in order to tackle those very important fundamental questions, I think the real frontier is beneath the sediment, the oceanic crust. The oceanic lithosphere has been largely unexplored yet by scientific ocean drilling, so we should do that in the future. So it has been observed, there are many evidences that microbial activities are going on within the oceanic crust, something like this. But first, direct evidence for microbial activity is reported from the Van de Fuck College, that is IODP Expedition 301, that I said with Mark Leber at ETH. And this is a 3.5 million year aged oceanic basalt, and we investigated the carbon and the sulfur isotope as well as DNA using those vein fractured the massive piece of basalt. And we observed some anomaly of sulfur isotope and carbon isotope, suggesting the occurrence of isotopic fractionation mediated by microbial ecosystem. And after almost 10 years, Shippo scientist of expedition C29 at the South Pacific region, the Yohei Suzuki at University of Tokyo, reported a fascinating finding in the communications biology 2 years ago. He discovered very dense microbial biofilm within the basaltic fracture. The basalt age is 100 million years. So that's the, and also there is no, it is impossible to occur the contamination because this fracture is filled with a non-tronite or some altered clay minerals. Okay. Then, okay, there are some possibilities that cluster biosphere is horizontally expand from the lit to open ocean. And what about spatial-temporal extent? Is there life in the deep oceanic crust and even in the upper mantle? Which is a simple but important question. The primary hypothesis is that in oceanic crust cooler than the summer limits to life, water-rock interactions sustain microbial life as deeply as seawater-derived fluid penetrates. So, if seawater penetrates into the rock, of course, water-rock interaction, including some radiolysis, produce some energy. So that the important thing is water. So if the water is available within the crust. In order to verify this, in my opinion, it is very important for human being to explore those environments because it's accessible. And of course, there's many unexplored realm beneath the seafloor, the oceanic crust down to the mantle. Of course, we need many technological development, but it's, I think it's possible in the future. I'm very much looking forward to realize this big project in the future. So I'd like to acknowledge my many colleagues all over the world and who sailed with me during the multiple expedition. And also I'd like to thank JDESC, DCO, and the Seafloor 40 program and the funding agencies, MEX and NSF. Thank you very much.